My clock says 10 a.m., so we'll get started. Thanks everyone for being here this morning. My name is Noah Newman, editor with No-Till Farmer, and welcome to this webinar, Practical Answers to Common Questions About Crop Decay with Midwest Biotech's Vice President, Doug Miller. Great to have everyone with us. So it can be highly beneficial to recycle valuable nutrients from crop residue due to the high cost and limited availability of fertilizers over the past few years. So Doug's going to share some valuable tips learned over the past several years, and he'll walk you through the many benefits you can achieve from accelerating your residue decay program. And before we get started, if you look at the bottom of your screen, there is a Q&A icon. That's where you could type in your questions throughout the presentation, and I'll see them on my screen. At the end of the presentation, we will have time for a Q&A session. I'll read some of those questions, and Doug will get you some answers. So let's get started. Doug Miller, take it away.
Thanks, Noah. Always good to be with the folks at Lessiter and with the no-till audience. I think this is number 11 for the webinars we've done, and we've already booked one for next year. So works well for us, and we always enjoy having an opportunity to talk about what's going on in the field and in our soil health lab and other places. We've talked about residue decay in several of our previous webinars. And as we've indicated with our title today, we're going to talk about some of the common questions we get. So we've covered a lot of the residue decay basics in our previous webinars. As Noah indicated, we've recently focused on recycling nutrients because of the high cost of commercial fertilizer and limited availability. So for the last few years, that's been the focus for a lot of people when it comes to residue decay. Longer term, we want to refocus on some of the other benefits, including building soil organic matter. We've talked about that. We've talked about who engages in residue decay and the fact that it's largely the fungal species. Bacteria play somewhat of a role, but anything you do to harm your fungal species in your soil is going to hamper your residue decay program. So reducing tillage is one of the big things that helps those fungal species. The difference between fungus and bacteria is that if you do something to harm the bacteria, they can usually regrow those colonies within a matter of weeks, but the fungal species are slower to regenerate and often take months to regrow. And consequently, anything that hampers those guys is going to slow down residue decay. And so we've talked about different strategies in the past for how to support those decay organisms out in the field. We've talked about the different types of residue and the fact that most of the attention's on corn residue because of the gross tonnage involved. Just rough rule of thumb, we've noted that each 1 foot of height for a corn plant generates about 1 ton of residue. Consequently, wheat and soybean residue don't get as much attention because you're dealing with about 3 tons of residue per acre on the surface relative to about 8 to 12 tons for corn. But in many cases, wheat straw and other small grain residues harder to break down than corn stalks on a per ton basis because it has more carbon and less nitrogen. The role of nitrogen is pretty important in residue decay because those decay organisms, as they ramp up and grow those colonies, they need nitrogen to form proteins and other biochemicals for their cell structure, just like we do. And they'll have to get that nitrogen somewhere. If it's limiting, that's going to limit the amount of decay activity in a field. And so consequently, relative to corn, soybean residue is easier to break down because it's got a lower carbon nitrogen ratio. It's got relatively more nitrogen in the structure of those stalks than corn, and also small grain residue has less nitrogen and more carbon. And so that stuff's often harder to break down on a per ton basis. So there's a critical role for nitrogen. And of course, the whole thing's driven by Mother Nature. So temperature and moisture play a key role. And we've talked about that in the past. Just from our last webinar, we showed some results from our soil lab where we used the Solvita burst test to measure CO2 carbon respiration from soil samples. And in the lab, we wet the samples to represent 60% of field capacity. That's kind of considered ideal for lab purposes. And microbial activity, if it's really dry, is going to be limited, and also if you've saturated that soil, aerobic microbes, those involved in the decay activity are going to be also limited. So there's that sweet spot in the middle where you've got some moisture in the field, but it's not saturated, where you're going to get the most activity. But if it's extremely dry, like it's been in a lot of the US this year, you're not going to have much decay activity. And also, if you saturate the soil, the same thing happens. It's also dependent on temperature. Now, this is air temperature. In the lab, we're incubating samples at constant temperature. They're relatively small samples. So the air temperature and the soil temperature are the same in the lab. And we incubate them at roughly 70 degrees Fahrenheit. And what we find is that you get peak activity somewhere around 85, 86, 87 degrees. But the key point is that if you've got really low temperatures or really high temperatures, that microbial activity drops off pretty fast. Out in the field, of course, you're going to have air temperature and soil temperature differences, but for that surface, the top 2, 3, 4 inches of soil, the cooler it is, the less activity you've got. And of course, if it gets really warm, there's going to be less activity. And so consequently, you'll have less decay activity late in the fall as that soil temperature cools, and it's going to ramp up again as spring arrives and you get to warmer soil temps, say April, May, June time. Microbes respond pretty fast to temperature changes. They can enter and return from dormancy in a matter of a few minutes. And so, for example, we were at the no-till conference last January in St. Louis, and the daily highs a couple of those days were over 50 degrees. And I know that there was some decay activity going on in the upper Midwest at that time because of those relatively warm temperatures, even in the middle of winter. So temperature matters. If it's too dry, too warm, too cold, we're going to have limited decay activity, and there's not much we can do. So it does depend a little bit on Mother Nature. So all of those things we've talked about in the past, and we'll leave you to the details in our previous webinars and our website and our newsletter and the other things we present. But today I just thought, since we're having a chance to refocus, we would consider some of the common questions that we get, mainly from farmers who are either customers or prospects that we talk to at farm shows, or they call our office, or we're standing out in somebody's farmyard kicking tires and talking over when we happen to be on the place. And there are a common set of questions come up, and they're all related to practical issues. And so we just thought we would take this opportunity to talk about some of these things that we do get as frequent questions, and we try to provide some practical answers to them. The first two questions are related. They're distinct questions, but the answers are related. The first one we commonly get is, hey, I am a strict no-tiller. I don't engage in any tillage. The only thing I do in terms of tillage is when I run my row cleaners on the planter. And otherwise, I'm not doing anything to stir the soil. Am I going to have problems decaying residue? Do you require soil contact in order to decay? Related question is, should I do something to shred my stalks? Vertical tillage tool to size the residue? Should I have a corn head that processes residue as it's moving through the field? Answer to both of those questions comes down to how the microbes colonize that residue when when the decay process starts. And the basic answer is the decay microbes are going to enter those stalks any place they can, any place that they can break into the center of the stalk and get at the easiest sugars to decompose, which is the pith or the cellulose in the center of the stalk. And that's true for corn residue, soybean stubble, wheat straw, whatever you have. The cellulose or pith in the center is the easiest to break down, and that's what they're going to go after first. As the picture we have here illustrates, this corn stalk has the most decay. It was first colonized at the top of the stalk where the snapping roll broke that stalk off as the combine moved through the field. That's where the organisms entered that stalk. That's where the most decay has happened. So the residue at that point is largely decomposed inside. What is left is sort of an ashy colored portion of the pith, and then they'll work down back toward the soil level from about a foot off the ground. So soil contact is not required. This particular field's in northern Illinois. It was a few years ago. The corn was harvested in early October. Producer applied our residue decay product roughly middle of the month, and then this picture was taken in late May. The beans were planted in early May, and they're coming up nice. There's plenty of residue there to provide cover for wind and water erosion protection. But if you look at those stalks, they are largely hollowed out. The cellulose is broken down, and then the residue decay organisms are working on the rest of that stalk. So once the beans are canopied, a lot of that stalk material is going to be decayed. At this point, the stalks are brittle on the outside. So even though they're present there as cover for the soil, protect against erosion, they're not going to cause any trouble with tires or planter openers or other equipment. And they're also have much diminished capacity to hold plant disease organisms or insects that could reside in there even over winter or into the spring. So to answer our two questions, no, you do not need tillage to decay residue. Like, you know, as illustrated here, the organisms are going to find their easiest way for a meal that they can find, even if it's well off the soil surface. Does it help to break those stalks in some way? Yes, it does. The more openings into that stalk that the organisms have, the more colonies they're going to be able to form, and you're going to break down those stalks in different and multiple places. So the decision to shred stalks or run vertical tillage or something else is up to you. That, you know, you have to look at your diesel fuel costs and your available time. Probably is going to be a year by year decision. But the short answer is if you do that, you can help with residue decay by breaking those stalks and providing openings for the colonies to start to form. A question we occasionally get from people when we talk about accelerating residue decay is, you know, why do it? And I've had people say, you know, gee, I got corn stalks out there. If I'm on continuous corn, I got residue left over from 2 or 3 years ago, and it's there. It doesn't. I run aggressive row cleaners. It doesn't cause me any trouble. I don't have trouble with tires. It's all good, and I view it as a savings bank. Why break it down right away? And it'll break down eventually. And so they view it as kind of like a savings account. If that's working for you, that's great. But what we find is there are a lot of advantages to removing that excess residue. One, the last couple of years, because of high commercial fertilizer prices, a lot of people have wanted to recycle those nutrients and get them back into production. Every fall and spring, we go and look at several different sources for commercial fertilizer, break them down on a per nutrient basis. And if you add up the value of 1 ton of corn residue, just last time we did it, it was about $40 per ton. So if you can break down another ton or two of residue, you can release some pretty valuable nutrients. Of course, that changes with commercial fertilizer prices. Like I say, the most recent estimate we had was about $40 a ton. And it varies by the type of residue. Even though you've got less soybean residue in the field, each ton is actually more valuable than corn stalks because of the additional nitrogen that's in soybean residue. They're usually about a couple dollars more per ton. And then small grain residues even worth more, usually about $7 or $8 a ton more than corn residue because of the higher potash content. So if you can recycle those things, that's to your benefit. Getting rid of that excess residue is also going to reduce problems with planting and emergence issues. And the one we hear most commonly is that residue can slow warming and drying of soils in the spring. It's going to push back your planting dates unless you can get rid of it. And then finally, we've seen more issues with not only insects, but especially plant diseases that are residue borne. And things like Gauss's wilt a few years ago, more recently tar spot. If you can reduce the habitat for those sorts of pathogens, you're going to have fewer problems. So it's a savings account in one way, but it tends to cause more trouble, and so we tend to find that it's better off to get it broken down. And of course, all of these comments are mainly focused on the northern half of the cropping areas in the U.S. As we've talked before, people down south don't have as many problems with residue because they've got fewer cold months in the winter to slow down the decay process, and their issues holding on to soil organic matter and not building it. And of course, the strategy there is you're going to have to supplement the active carbon in the soil by using covers and other methods because you can't hold on to the soil organic matter. But those of us in northern areas that have winter to deal with and have slower residue breakdown, anything you can do to accelerate that process is going to help you avoid some of these problems over time. Very common question is related to the role of nitrogen in the decay process. Some people tell you, hey, to decay residue, you got to add nitrogen. Our answer to that is it depends. As we've already indicated, the microbes do require nitrogen for their cell structure. It's basically an 8-to-1 ratio. For each 8 units of carbon they're going to break down, they're going to need about 1 unit of nitrogen just to form those microbial colonies in the decay organisms. They're going to get it from any source. And while that decay process is underway and those colonies are ramped up, you're going to tie up nitrogen in that microbial biomass. This funny looking image here at the bottom of the screen is what we call the residue decay smile. We've used it to illustrate this point for many years. The brown portion of it represents the decay activity. So as it starts out on the left side relatively low and then ramps up, those microbial colonies increase in number. And as they do, they're going to decrease the available nitrogen available in the field. And that's represented by the green portion of the diagram. So at peak decay, you're going to have the least amount of nitrogen available because it's tied up in that microbial biomass and won't be available for growing a crop. So the last thing you want to do is to have your peak residue decay occurring at a time when you're also wanting to use that nitrogen and other nutrients for your cash crop. And so it helps to accelerate that process, get it over and done with before you want to use those nutrients for other things. Rough rule of thumb, for corn stalks, you're going to need about 13 pounds of nitrogen to decay an additional ton of crop residue. So if you get a decent fall, you got about 6 weeks of relatively warm weather, you've got ample moisture, you might decay an additional 2 tons of residue, and it's going to take somewhere between 25 and 30 pounds of nitrogen to do that. Most cornfields at the end of the season are going to have that available. And so if you've got that much N available for your microbial colonies to ramp up and start the decay process, you don't really require any additional nitrogen. The it depends part of that comes under certain situations where that nitrogen is going to be short. Most common situation we see is cases where you've had very heavy rainfalls sometime during the season and you're going to leach nitrogen so that you're more or less running on empty by the end of the season after you've completed the crop and hauled away the grain. There may not be enough additional nitrogen left over to decay residue at the rate you want. It's going to happen more slowly if it's limited by nitrogen. The other case, and this is more common, is people have adopted cover crops. If you're starting fall rye or some other grass cover that's going to use a fair amount of nitrogen in its early stages, and you're trying to decay residue, you could end up short. And as this picture illustrates, I've seen several fields over the past few years where somebody has a nice rice stand started, cover crop got off to a good start, germinated well, but it just turns yellow after time. It can turn yellow for several reasons, but a lot of times if you look at the leaf structure and it's the lower leaves that turn yellow, that's a symbol that, or a sign that it's probably short on in. And so if you're going to start a rye cover and grow at 6-8 inches in the fall, if you get it started early enough, that's going to take somewhere between 20 and 35 pounds of nitrogen. And you're trying to decay residue at the same time, you might be looking at somewhere 50, 60 pounds of N required. And if you don't have that much out there, you're asking your nitrogen to do double duty, and you're going to slow down your residue decay and harm that cover crop, or at least slow its progress. And so some additional nitrogen may be required if you're going to put on a fall grass cover and want to get it started. So there is a role for nitrogen. It isn't always required, but if you're in one of these situations, it's definitely something you want to consider. One of the biggest issues we've noticed over time, and it first happened on our farm, you can decay weed seeds. They're crop residue just like stalks and cobs and leaves. And over time, it is possible to reduce weed pressure through an active decay program. As I say, on our farm, this was one of the first things my dad noticed. He got involved in biologicals in the late 70s, and I think the first residue decay tests were done 79 or 80. And a few years later, he'd noticed that we had a lot less shatter cane in the field where the tests were conducted. Now, shatter cane's a warm season grass. It has been introduced for a long time and still in use as a forage grass. My grandfather raised cattle, tried shatter cane as a forage source for his livestock operation, and then moved away from it pretty quickly because under the wrong conditions, it can either generate cyanide or nitrate toxicity, which is very harmful to the cattle. And so he decided not to do that anymore. But those seeds persist for a long time. Shatter cane's an annual, but it regrows from its own seeds. Each plant can grow several hundred seeds, and they're fairly persistent in the soil. So the shatter cane in that one field had been a problem for many years, and even in the roughly 1980, there was still a lot of weed pressure from it. And my dad noticed that there was getting to be less. We've had people notice similar things in their own fields over time, whether it's tough weeds like Johnson grass or its cousin shatter cane, Jerusalem artichokes, and more recently, things like Palmer amaranth, pigweed, and its related cousins. The role of residue decay in affecting the weed seed bank is not terribly well understood, to be quite honest. The microbial activity and its impact on the weed seed bank is something that the agronomists tell us that they don't know quite as much about. They do know that the weed seed bank is affected by the type of dormancy that the seed goes through. Some seeds don't ever really enter dormancy, and if once they're mature and they fall off the plant, they can regrow almost immediately. Other seeds will have to go through a cool period or be exposed to moisture or a dry storage period, and then they can regenerate. Persistence depends on many factors, but probably is most correlated with the seed coat. So The nice thing about pigweed and palmer and water hemp is even though each plant generates 50 to 70,000 seeds, if it's a mature plant, the seeds are relatively small and they've got very thin seed coats, and so they'll decompose pretty quickly. Other seeds like velvet leaf have very thick, hard seed coats. The seeds themselves aren't very big, but that seed coat's really tough, and that stuff can persist in the soil for decades. I was on a farm not too long ago where somebody had pulled up an old feeding floor or part of a driveway or something, had been covered in concrete for years, and shortly after that ground was now exposed to the elements again, they grew a pretty nice crop of velvet leaf. Yeah, it might have blown in, or it might have been carried in by birds, but the fact that it was more or less limited to the area where this old concrete pad had been indicates that it very likely grew from seed that had been there since the concrete was formed. That those seeds can last for decades, and there are some seeds that will persist for 75 or 100 or more years in the soil. Takes a really long time to get them out of dormancy. Dormancy, other than cool, wet, dry cycles, can be alleviated in some cases by fire. I saw a TikTok not too long ago where somebody here in Illinois had an old grass pasture that really hadn't been used or managed for long. They just mowed it once or twice a year, keep the weeds down. But they had ultimately decided to regenerate it as native prairie, and the first thing they did was burn it. And once the pasture recovered from the burning process and things started to regrow, they noticed species that they hadn't seen in years, plants that hadn't really been growing there in anybody's lifetime. And it's quite likely that those seeds had been brought out of dormancy by the fire. And so there are lots of things that affect dormancy and persistence in the soil, and that's why you tend to see weeds that last and come back years and years and years, even if you bury them with tillage, bring them back up. Many years later, they'll regenerate. And then the role of microbes with the weed seed bank is not very well understood. Interaction between microbes that are pathogens and seeds is pretty well understood because of all the testing done for commercial fungicides and other chemical seed treatments. The role of microbes, especially bacteria and inoculant treatments, is reasonably well understood, but the role that microbes play in decay of seeds is not well understood at all. And that comes from the researchers in the agronomy literature. They'll admit that there's a lot of things that happen that they don't know about. We've all heard about root exudates thanks to the folks at Lesidure and the no-till conference, and you hear about it at the strip-till conference and other places. One of the many benefits of cover crops is that those root exudates help feed your microbial populations under the soil. Well, seeds exude chemical properties, compounds, and some of them are relatively high in sugar and attract one set of microbes. Others of them are relatively low in sugar or carbohydrates and attract different microbes. And it appears that in some cases that helps to protect the seed by, for example, growing microbial or bacterial colonies near the seed that help fend off fungal infections. But in terms of decay, not much is understood. The agronomists know that it happens. They've done a lot of studies over the years where they've studied the impact of dormancy by actually burying groups of seeds in soil and then going back periodically to count, how many have germinated, how many have come out of dormancy but haven't germinated yet, how many have entered a secondary dormancy because conditions weren't quite right, maybe not enough moisture, maybe the temperature changed, and then count the seeds that are no longer viable and basically are dead and won't regrow. And then there's a gap. They know that some have decayed, but the process by which that happens isn't terribly well understood. There are different theories about why it happens, but there haven't been a lot of tests done. From the empirical side, actual measurements of this process, people have found that you can decompose somewhere between 20 and 80% of particular weed seeds over the course of a couple years' time. So it doesn't happen right away, but you can use biological decay to help eat into that seed bank. The species that decompose more are those like palmer and pigweed that have relatively thin seeds, easy to break down. Things like velvet leaf and native grasses and other things that have harder seed coats, you can decompose, but it happens at a lower rate. And so the agronomy research has shown that the decay rates depend largely on how tough those seeds are. But it takes time. You're not going to get a perfect control, but you can get some reasonable control by encouraging microbial decay of your seed bank. The other thing they've noticed is based on different tillage practices, no-till versus conventional tillage, and things like that, that soils that are more biologically active tend to have higher decay rates. So anything you can do to promote biological activity in the soil is also going to promote those decay organisms, and they're going to have a better chance at eating into that weed seed bank. And as I say, we've had customers notice this. We noticed it ourselves on our own farm. It's hard to measure, but people notice it. We've had customers in Central Illinois who've worked on their decay programs for years, and they haven't completely eliminated water hemp from their fields, but they've got noticeably less than neighboring fields and less than they used to have. Another area where we've done probably the most work is on volunteer corn. Corn ears, when they mature, those kernels typically are dormant when they're harvested, and then they've got to go through a cool cycle to bring them out of dormancy so that when you plant seed corn in the spring, it's ready to go. While it's dormant, it's not going to be able to grow. And so if you can decay those ears, if they're dropped from the combine or they're dropped when the corn head's moving through the field, they'll generate into lots of volunteer corn unless you can decay them. And just this picture, it's a little bit fuzzy, but it shows a partial ear. It's probably got 300 or 400 kernels on it. Some of the kernels are laying right on the soil surface and would be subject to germination, but they were decayed before, and only one kernel out of the hundreds actually germinated here in the lower right. So if you can work on your residue decay program, break down those kernels, at least decay the germ on those kernels enough so that they cannot regrow, they're no longer viable seeds, you're going to have a lot less volunteer corn problem. And that's something else that people have noticed over time. After the derecho a couple of years ago, we had a increased use or interest in residue decay programs because people dropped a lot of corn ears due to the windstorm, and we had quite a bit of success. We had relatively clean bean fields in some areas, even though the number of ears on the ground was large. You went out and counted them. They were in many cases moldy or decayed, looked very much like this one, but little to no germination if you can get those broken down fast while they're still dormant. Last major question and this is one that's come up a lot recently, especially as people are pushing the season on early planting and increased interest in cover crops. Hey, if I'm trying to work on residue decay at the same time I'm planting earlier, or I'm trying to get fall covers in, am I shooting myself in the foot by trying to put new seeds out there at the same time I'm trying to decay crop residue? You just said that seeds are part of crop residue, and am I going to decay those faster than they germinate, so that I don't get the stand. I'm not going to get what I'm paying for in new seed, and I'm going to be relatively unhappy with the results. Again, I'll refer more to the science this time than to our experience. Our experience is that that's not a problem. We haven't seen any cases where we felt that that was an issue. But to be more reliable in providing an answer to this, we'll go to the science, the agronomy research. And the agronomists point out that one difference between weed seeds and crop seeds is that crop seeds tend to have more limited dormancy character than weed seeds, mainly because of the genetic selection process. Crop seeds have been selected for certain genetic attributes, including ease of germination and early seedling vigor. As a result, the seeds that we're now planting, the germplasm that's currently being used for row crop seeds, corn, wheat, soybeans, etc., is going to have fairly limited dormancy that's fairly easy to break, and that helps with that ease of germination and early vigor. As an example, the agronomists point out that this selection process has led to side effects like pre-harvest sprouting. It's not uncommon in wheat to see wheat kernels sprout on the heads before they're harvested. I've seen this myself in corn. It's pretty rare, but it has happened. I think 6 or 7 years ago, we had September that was really wet, very warm, very humid. And we were in a couple of fields that fall early and saw corn kernels sprouted on the cob, on the stalk before harvest. So the kernels were physiologically mature. It wasn't dry enough to harvest yet, so it was still on the stalk. And they were not dormant because they were starting to sprout right there on the cob. As I say, typically corn kernels are dormant at physiological maturity, but not always. And it's pretty easy to break dormancy because of the selection process. And so it is possible to see wheat kernels and corn kernels that sprout before they're even harvested. The researchers point out that this is less common in soybeans. I guess theoretically it can happen, but it's not something we see often because the soybean seeds have a harder seed coat. That waxy fingernail-like material that forms the soybean hull is harder to penetrate with water. It's more durable. And so even under the same conditions where you get pre-harvest sprouting on wheat or corn, you tend not to see it on soybeans because those kernels are protected by that harder seed coat. Bottom line from the research, at least on this line, is that seeds you're planting have been brought out of dormancy. They're ready to go. And they're typically going to germinate faster than they would decay. You use seed treatments, fungicides, and other things to protect them against pathogens, but the decay organisms are different. They can decay seeds in dormancy, but typically once they're out of dormancy and you've planted them not always in ideal conditions, but under favorable conditions, they're going to grow faster than they would ever decay. The only real risk of decay is if the conditions change, weather really dries or gets cold, and those seeds go back into secondary dormancy and then sit there for a while. That's when the decay can happen. But the other thing is that if you have dry, cold conditions that aren't going to help germination, you're also going to have slower residue decay. If the seeds sit there long enough and then decay, you're talking about a replant situation anyway. But the bottom line is that based on the selection process, your decision of when to plant and the nature of those seeds, they're going to usually germinate way faster than they decay. And once they're germinated, then the decay organisms aren't going to affect them. Pathogens might get them, but not decay organisms. And so that's our short answer. And again, it relies on what the agronomy literature is telling us, and our experience is consistent with that. We don't usually see problems with residue decay affecting growing seeds or plants or germination, things like that. So with that, Noah, we'll open it up to any questions.
Unmute my mic there. Thank you very much, Doug. Great presentation there. Yeah, if you have any questions right now, bottom of your screen, you'll see the Q&A icon there. It's right next to the raise hand icon. And type in your question there. I'll see it pop up on my screen here, and we'll get those relayed to Doug. Also, quick reminder that the entire webinar, we're recording it, and we'll be publishing it, posting it to our website, no-tillfarmer.com, emailing it to your inbox as well. So the recording of the webinar should be available within a couple days usually. But as we wait to get some questions here in the inbox, Doug, I've got one for you. Are there any tips or keys to success to make sure residue is spread evenly across the field so that you don't have it spread unevenly?
Yeah, that's getting to be a real problem. As combine headers get bigger, we've had more people complain that even though they've got straw choppers and other equipment to distribute in the back of the combine, that it just can't spread it as wide as they would like and as evenly. And so some people are going out and having to disc it or rake it or do something. And we have had people, especially the biggest problem is in double cropped wheat and soybeans. So you harvest the wheat crop and you leave even if you don't mean to, you leave a windrow behind the combine, and that can hamper not only the planting but the emergence of the following bean crop. And so we've had cases where people have applied our residue decay product or others to try to, they just put it on the windrow to help break that stuff down so it's not going to cause a problem. The good news is that at the time of the year you're doing that, you've got plenty of temperature and usually ample moisture, other than years like this, to help encourage that decay. So, but that's an issue. I always go back to the very first time I noticed residue decay products working. Was on our own farm again. I was in high school when my dad got involved in this, and so in the spring of the year, I was sent out with a field cultivator to work the field ahead of the planter. And I got we had one low spot in the corner of the field, and if we had a heavy rain early in March that year, and a lot of the stalks washed into that low spot, and that can be a good recipe for plugging up a field cultivator very quick. Usually you'd have to go through it 4 or 5 times, stop, shake out the field cultivator, go back, hit it again. And I was not around at the time they put out the residue decay treatment in the fall, and so I'd kind of forgot about it. I come up over the rise, getting ready to go down into this low spot, and I saw those stalks, and I thought, oh, blank, fill in your favorite expletive. And this is going to be trouble, but went through it pretty quickly and didn't even have to go through it a second time. And it's because those stalks were decayed enough that they broke rather than clogging up the implement. Of course, we weren't no-till at that time, so we were running a field cultivator. In a no-till situation, that can be a real problem, but anything you can do to help break down those stalks, even if you get a thick mat, they'll be there for cover, but if they're hollow and the outside is decomposed enough to be brittle, you can go through it with planting equipment and not cause trouble with tires and other things.
All right, we have several questions pouring in. How will a legume cover crop change available in a positive way?
Yeah, legume cover crops are great because they will help provide additional nitrogen. Depends on the legume cover crop you're using, but that will be a positive for residue decay because one, they require less nitrogen to get started generally, and two, they're going to contribute whatever nitrogen they fix and make it available for the following crops. So yeah, legume covers are nice. People like rye and other grass covers because they're fast and cheap. Um, but the other types of covers do have their benefits.
How do fall applied fungicides affect residue breakdown?
Well, we don't do research on fungicides ourselves, but I'll refer back to the no-till conference. I think it was 6 or 7 years ago. There was a breakfast sponsored by one of the chemical companies, and that question came up in general forum. One of the members of the audience asked the research director, hey, if I'm applying fungicides, how does that harm or help or affect my soil health? And they didn't quite really directly answer the question, but there was an agronomist in the very next session, a university researcher who said, I can answer that, and said that the fungicides are going to be harmful not only to the pathogenic fungus, but also the beneficial ones. And so if people ask us about, use of fungicides, and we say, if you have a particular problem, you better use it and, get that under control. But in general, we find that people get away from it if they don't have a specific problem. So, for example, this year being drier, relatively cool, low humidity conditions here in the upper Midwest, there were fewer fungal problems that at least that we've seen, and you could probably get away without a fungicide and not just do it as routine practice. If you've got a specific problem, then yeah, go ahead and do it, but you'll have to recognize that you may be hurting those fungal populations, including the decay organisms. And so then if you can do something to help regrow those guys, you'll avoid that that loss that the chemical treatment is going to impose.
All right. Someone wants to know if you could repeat the info about how much nitrogen per amount of corn is needed for residue decay.
Okay, yeah, and this is just a real rough rule of thumb. This is kind of a static estimate. It's not based on the dynamics of the process, but we figure roughly 13 pounds of N per ton of residue decayed. And so the example I gave was, if you harvest early enough that you've got about 6 weeks of decent weather before things freeze up, you might be able to decay about 2 tons of residue. And so that means about 26 pounds of N, so roughly 25 to 30. And most cornfields are going to have that left over at the end of the season. So in that case, and you've got another special circumstance, you probably have enough nitrogen out there to handle the amount of residue you're going to decay yet that fall season. If you're harvesting later, so your decay window is shorter, you're going to use less nitrogen. If you're harvesting some places late August, early September, that happens around here if things get planted early and you have the right year, you may decay 3 or 4 tons of residue in the fall under the right conditions, and you actually may need a little bit more nitrogen than that. But that's the rough rule of thumb that we use. And as I say, if you think you're going to be short of roughly 25, 30 pounds of available N in the field, that's when you may want to consider putting on some fall nitrogen, especially if you lost some to leaching due to heavy rainfall during the season, or if you're raising grass covers that require more nitrogen and you're asking the N in your field to do double duty, it may need some help. And to get the cover started and growing in the way you want and to decay that residue, you might need another 30 pounds of N.
Okay. And a reminder, if you have any questions, now's the time to get them in the Q&A icon there in the bottom of your screen. We have someone who would like you to talk a little bit about the Midwest Biotech Chandler product, and looks like they're curious about that. If you want to touch on that a little bit.
Yeah, we try not to make this a sales pitch as much as an informational pitch, and that's what we've always done with our webinars. But just very quickly, you can go to our website, midwestbio-tech.com. All the information's there. But briefly, we sell a product called Biocat 1000, and unlike a lot of other decay products on the market, it's enzyme based, which means it's intended or designed to multiply the decay organisms already in your field. Does not have any live fungus or fungal spores or bacteria in the container. The benefit of that is the product has a long shelf life. We usually tell people 4 years. So if you would buy some for fall and you don't get it on because it gets late or it's too wet or too something, you can put it on early in the spring. You could put it on next fall and it would be just as good. Also, since there's no microbial content, you could put it on with fall burn down chemicals. You can put it on with liquid 28 or 32, other liquid products. And since you're growing the microbes that are already present in your field, you don't have chemical compatibility issues. Sometimes some cultured microbes were cultured in one pH environment, and if your soil pH or your soil chemistry is a little bit different, they may not thrive quite as well. So this product's designed to work on the microbes already present living in your soil, just help them live, multiply, and do their job. So that's kind of what we've seen. We've worked with these products now, as I say, since about 1978 or 79 or 80. We did 4 years of tests on our own farm before we started marketing in 1981. So we've been at it selling for 42 years, but actually longer term experience. And as I say, I was in high school when my dad got involved in this. So I was the guy out in the field carrying the spade and counting the plants and drop ears and all that sort of fun stuff. And so we've got quite a bit of experience with it. And we've stayed with the Chandler products for many, many years because they're cost effective, they're reliable, they're a good company to work with. And we believe our customers find the same thing is true. So if you want more details about the products, you can find that on our website.
All right. Well, we have time for a few more questions. I don't see any right now, but I'll ask you this, Doug. So how long does it take? So if you just recently switched to no-till, I mean, how long does it take to build up that soil biology and those microbes that really help with residue breakdown?
I would expect, and this is based on our experience, and as well, I hear similar things from people that have made that transition at the no-till conference. I would expect somewhere between 3 to 5 years. It depends on what your soil's like going into the process. If you've been heavy tillage, lots of chemicals, high salt fertilizers, it may take a while because those microbial populations have been under stress due to high tillage, wrong soil chemistry, all that sort of thing. And you hear growers talk about this that aren't involved in biological products but are just making the transition by changing their tillage practices. It takes a while. Now, if you've been working on your soil biology, you've been applying manures and reducing tillage, using covers, the transition's much easier because you don't have to dig yourself out of as big a hole. To be kind of blunt about it, we've seen cases where people have fairly active soils, even though they're using conventional tillage practices, and they just help themselves by reducing the amount of tillage that they do. But that transition will be on the shorter side if you've been doing other things to help yourself before you make that no-till transition.
Okay, well, I don't see any more questions at the moment. Any last words of advice you want to give to our viewers right now as fall is right around the corner here?
Yeah, I guess the thing, if you're going to use our product or somebody else's, or just make some change to help your residue decay program, the thing we say is earlier the better. When you apply our Biocat, if you can get it on as soon after harvest as possible, that usually helps. But the last few years have been fairly late falls, late planting, wet in the fall, things didn't dry out. And so a lot of times people don't have opportunities to apply product in the fall. And we got a lot of guys just honestly say, hey, I don't want to dig my sprayer out of the shed and haul a lot of water in late October, early November. So I'm going to do it in the spring. And we've actually had really good results with spring applications. As early as mid-March up through first week of May. The benefit of a spring application is usually you've had some winter moisture to help the moisture situation. So even if it's dry in the fall, usually you're going to have overwinter moisture to help. Doesn't always happen, but usually does. And you've got that ramp up in temperatures as you move into the spring. And some of the best decay we've seen is, late May, early June, where you've got ideal moisture, you've got daily high temperatures in the 80s, and you can decay a lot of residue pretty fast in a few weeks during that time right after planting. So even late applications will be helpful in a crop season.
Okay, thanks so much again, Doug Miller, for your time today. And again, what's the website if people want more information?
MidwestBio-tech.com is our main website, and we've got product brochures, we've got field results, all of our pricings there. We put out a quarterly newsletter, have since 1990, and the newsletters from the last 3 years are all there, have all of our latest information and field results and things like that. So, and if not, send us email or give us a call. We always answer our phone.
Sounds great. All right. Well, on behalf of No-Till Farmer, Midwest Biotech, and Doug Miller, thank you all for tuning in. Thank you all for tuning in today to this webinar. And we will have the recording sent to your email inbox and posted on no-tillfarmer.com and the No-Till Farmer YouTube page as well in the coming days. So be on the lookout for that. And if you have any ideas for future webinar topics you'd like to see covered, shoot us an email as well. Okay, thank you very much. Everyone have a great day.